Getchi.
Hello, my name is Nimari Getchi.
Hello, my name is Shona Getchi, and we're presenting the basic points in inflammation and atherosclerosis.
Atherosclerosis. Atherosclerosis is a specific form of atherosclerosis in which an artery wall thickens as a result of invasion of accumulation of white blood cells and proliferation of intimal smooth muscle cells, creating a fibrofatty plaque. The accumulation of white blood cells is termed fatty streaks early on because of the appearance of being similar to that of the marbled state. These accumulations contain both living, active white blood cells and remnants of dead cells, including cholesterol and triglyceride. Atherosclerosis. Atherosclerosis is the thickening, hardening, and loss of elasticity of walls of arteries. This process gradually restrains the blood flow of one's organ and tissues and can lead to severe health risks brought on by arteriosclerosis, which is a specific form of atherosclerosis caused by the buildup of fatty plaques, cholesterol, and some other substances in and on the artery walls. Treatment is often a form of preventive measures of prophylaxis. Lipoproteins. A lipoprotein is a biochemical assembly that contains both proteins and lipids, which allows fats to move through water outside cells. The proteins serve to emulsify the lipid molecules to create a complex particle which is both water soluble and can transport all fats within. Many enzymes, transporters, structural proteins, antigens, adhesins, and toxins are lipoproteins. Examples include the plasma. Lipoprotein particles classified under HDL, LDL, IDL, VLDL, and LDL. Monocytes. Monocytes are a type of white blood cell or leukocyte. They are the largest type of leukocytes and can differentiate into macrophages or dendrite cells as a part of the vertebrate innate immune system. Monocytes also influence the process of adaptive immunity. Monocytes are produced by a bone marrow from precursors called monoblasts, biopotent cells that differentiated from hepatomotic cells, stem cells. Monocytes circulate the bloodstream for about 1 of the 3 days and can typically move into the tissues throughout the body. LDL, low-density lipoprotein, is one of the 5 major groups of lipoproteins. These groups, from least dense to most dense, are cyclomicrons, very low-density lipoproteins, intermediate density lipoproteins, low-density lipoproteins, and high-density lipoproteins. Lipoproteins transfer lipids around the body in the extracellular fluid. They can be sampled from blood and allow fats to be taken up by the cell of the body by receptor-mediated endocytosis. Lipoproteins are complex particles composed of multiple proteins. HDL, high-density lipoproteins, are one of the 5 major groups of lipoproteins. Lipoproteins are complex particles composed of multiple proteins which transport all fat molecules around the body within water outside cells. They are typically composed of 80 to 100 protein per particle. Increasing concentrations of HDL particles are strongly associated with decreasing accumulation of atherosclerosis within the walls of arteries. This is important because atherosclerosis eventually results in sudden plaque ruptures, cardiovascular disease, stroke, and other vascular diseases. Artery wall. Arteries are blood vessels that carry blood away from the heart. While most arteries carry oxygenated blood, there are 2 exceptions to this: the pulmonary and umbilical arteries. The effective arterial blood volume is the extracellular fluid that fills the arterial system. The outermost layer is known as the tunica externa, also known as the tunica adventita, is composed of connective tissue made up of collagen fibers. This is the structure of the artery wall.
Cholesterol is an organic molecule. It is a sterile or modified steroid, a lipid molecule, and is biosynthesized by all animal cells. Because it is an essential structural component of all animal cell membranes that is required to maintain both membrane structural integrity and fluidity. According to the lipid hypothesis, since cholesterol, like all fat molecules, is transported around the body in the water outside cells, inside lipid protein particles, elevated cholesterol concentrations, hypercholesterolemia, potentially offers a lower cost way to estimate concentrations of LDL particles, possibly even low concentrations of functional HDL particles. Both variations strongly associated with cardiovascular disease because LDL particles promote atheroma development in arteries. Cholesterolemia and mortality shows increasing threat of atherosclerosis as cholesterol in blood increases in relation, resulting in heart attack and death. Endothelial cells. Endothelium is a type of epithelium that lines the interior surface of blood vessels and lymphatic vessels, forming an interface between circulating blood or lymph in the lumen and the rest of the vessel walls. It is a thin layer of simple squamous cells called endothelial cells. Endothelial cells in direct contact with blood are called vascular endothelial cells, whereas those in direct contact with lymph are known as lymphatic endothelial cells. Endothelial dysfunction, or the loss of proper endothelial function, is a hallmark for vascular diseases and is often regarded as a key early event in the development of atherosclerosis. Impaired endothelial function causing hypertension and thrombosis is often seen in patients with coronary artery disease, diabetes, mellitus, hypertension, and hypocholesterolemia. Macrophages. Macrophages are a type of white blood cell that engulfs and digests cellular debris, foreign substances, microbes, cancer cells, and anything else that does not have the types of proteins specific of healthy body cells on its surface in a process called phagocytosis. Macrophages are the predominant cell involved in creating the progressive plaque lesions of atherosclerosis. Different types of macrophages. Both M1 and M2 macrophages play a role in promotion of atherosclerosis. M1 macrophages promote atherosclerosis by inflammation. M2 macrophages can remove cholesterol from blood vessels, but when the cholesterol is oxidized, the M2 macrophage becomes apoptotic foam cells, contributing to the atheromatous plaque of atherosclerosis. Lipid oxidation, otherwise known as lipid peroxidation, is the oxidative degradation of lipids. It is a process in which free radicals steal electrons from the lipids in cell membranes, resulting in cell damage. This process proceeds by a free radical chain reaction mechanism. It most often affects polyunsaturated fatty acids because they contain multiple double bonds in between which lie methanol bridges that possess especially reactive hydrogen atoms. Antioxidants. An antioxidant is a molecule that inhibits the oxidation of other molecules. Oxidation is a chemical reaction that can produce free radicals, leading to chain reactions that may damage cells. Antioxidants such as thiolose or ascorbic acid, vitamin C, terminate these chain reactions. Oxidative stress is thought to contribute to the development of a wide range of diseases. In many of these cases, it is unclear if oxidants trigger the disease or if they are produced as a secondary consequence of the disease and from general tissue damage. One case in which this link is particularly well understood is the role of oxidative stress in cardiovascular disease. Here, low-density lipoproteins, otherwise known as LDL, oxidation appears to trigger the process of atherogenesis, which results in atherosclerosis and finally cardiovascular disease. Platelets. Platelets, also called thrombocytes, are a component of blood whose function, along with the coagulation factors, is to stop bleeding by clumping and clotting blood vessel injuries. Platelets have no cell nucleus. They are fragments of cytoplasm that are derived from megakaryocytes. In addition to being the cellular effector of hemostasis, platelets are rapidly deployed to sites of injury or infection and potentially modulate inflammatory responses by interconnecting with the leukocytes by secreting cytokines, chemokines, and other inflammatory mediators. Collagen. Collagen is the main structural protein in the extracellular space in various connective tissues in animal bodies. As the main component of connective tissue, it is most abundant protein in mammals, making up from 25 to 35% of the whole body protein content. Depending upon the degree of mineralization, collagen tissue may be rigid, compliant, or have a gradient from rigid to compliant. Collagen in the form of elongated fibrils is mostly found in fibrous tissue such as tendons, ligaments, and skin. Throughout the 4 phases of wound healing, collagen performs the following functions in wound healing. Guiding function. Collagen fibers serve to guide fibroblasts. Fibroblasts migrate along a connective tissue matrix. Chemotactic properties. The large surface area available on collagen fibers can attract fibrogenic cells which help in healing. Nucleation. Collagen in the presence of certain neutral salt molecules can act as a nucleating agent, causing formation of fibril structures. A collagen wound dressing might serve as a guide for orienting new collagen deposition and capillary growth. Hemostatic properties. Blood platelets interact with the collagen to make a hemostatic plug. Extracellular matrix. In biology, the extracellular matrix, otherwise known as the ECM, is a collection of extracellular molecules secreted by cells that provides structural and biochemical support to the following cells. Because multicellularity evolved independently in different multicellular lineages, the composition of ECM varies between multicellular structures. However, cell adhesion, cell-to-cell communication, and differentiation are common functions of the ECM. Collagen are the most abundant proteins in the ECM. In fact, collagen is the most abundant protein in the human body and accounts for 90% of bone matrix protein content. Collagens are present in ECM as fibril proteins and give structural support to resident cells. Collagen is excised in precursors in the form of cleaved procollagen to allow extracellular assembly. Disorders such as Ehlers-Danlos syndrome, osteogenesis imperfecta, and epidermolysis bullosa are linked with the genetic defects in collagen encoding genes. Subendothelial space. The chronic endothelial injury hypothesis is one of 2 major mechanisms postulated to explain the underlying cause of atherosclerosis and coronary heart disease. Ross and Glossman initially proposed that endothelial cell uncovering was the first step in the development of atherosclerosis. Other hypotheses have associated the role of infectious agents in inflammatory responses in the artery wall. Currently, most research seems to focus on inflammatory processes which associate endothelial dysfunction with lipoprotein accumulation. The first phase of the inflammatory process is marked by the accumulation of lipid and low-density lipoprotein particles beneath the endothelium. This is followed by the attachment of leukocytes, monocytes, and T lymphocytes to the endothelial cell surface. Leukocytes migrate to the subendothelial space and aggregate within the intima. Chemotaxis. Chemotaxis is the movement of an organism in response to chemical stimulus. Somatic cells, bacteria, and other single-cell or multicellular organisms direct their movement according to certain chemicals in their environment. Leukocyte adhesion. Leukocyte adhesion deficiency, otherwise known as LAD, is a rare autosomal recessive disorder characterized by immunodeficiency resulting in recurrent infections. LAD was first recognized as a distinct clinical entity in the 1970s. The classic descriptions of LAD include recurrent bacterial infections, defects in neurotrophil adhesion, and a delay in umbilical cord sawing. The adhesion effects result in poor leukocyte chemotaxis, particularly neutrophil inability to form pus and neutrophilia. MCP1. The chemokine Lingad2 is also referred to as monocyte chemoattractant protein, otherwise known as MCP1, and a small indocible cytokine A2, CCL2, is a small cytokine that belongs to the CC chemokine family. CCL2 recruits monocytes, memory T cells, and dendritic cells to the sites of inflammation produced by either tissue injury or infection. CCL2 is implicated in pathogenesis of several diseases characterized by monocytic infiltrates, such as psoriasis, rheumatoid arthritis, and atherosclerosis. APOA1. Apolipoprotein A1 is a protein that in humans is encoded by the APOA1 gene. It has a specific role in lipid metabolism. The text in a recent report suggested that APOA1 mRNA is regulated by endogenously expressed antisense RNA. The protein promotes fat efflux, including cholesterol from tissues to the liver for excretion. It is a cofactor for lecithin cholesterol acetyltransferase, which is responsible for the formation of most plasma cholesterol esters.
Phospholipids. Phospholipids are a class of lipids that are a major component of all cell membranes. They can form lipid bilayers because of their amphiphilic characteristics. The structure of the phospholipid molecule generally consists of two hydrophobic fatty acid tails and a hydrophilic phosphate head, joined together by a glycerol molecule. The phosphate groups can be modified with a simple organic molecule such as choline. A triglyceride is an ester derived from glycerol and three fatty acids. They are also present in the blood to enable bidirectional transference of adipose fat and blood glucose from the liver and are a major component of human skin oils. There are many different types of triglyceride, with the main division being between saturated and unsaturated types. In the human body, high levels of triglycerides in the bloodstream have been linked to atherosclerosis and by extension the risk of heart disease and stroke. However, the relative negative impact of raised levels of triglycerides compared in that of LDL and HDL ratios is yet unknown. The risk can be partially accounted for by a strong inverse relationship between triglyceride level and HDL cholesterol level. D4F. Cholesterol efflux is the key step of reverse cholesterol transport and has become a therapeutic target against atherosclerosis. Human apolipoprotein A1 is the main protein in high-density lipoprotein that has 243 amino acids. APOA1 mimetic peptides have no sequence homology to APOA1, but possess the class A helic motif that presents an APOA1 lipid binding domains. D4F is one of the APOA1 mimetic peptides and exerts diverse arthroprotective functions similar to APOA1. L4F. The apolipoprotein A1 mimetic peptide L4F is a putic anti-diabetic drug, and it has antioxidant and anti-inflammatory properties and improves endothelial function. The platelet actives of L4 can prevent heart and coronary dysfunction in the mouse model of genetically induced type 2 diabetes. L4F increases insulin sensitivity in adiponectin levels while lowering inflammatory cytokines. A reduction in atherosclerosis with L4A1 was recorded. Further studies on the effect of APO1 mimics on atherosclerosis and insulin resistance in a variety of dietary contexts are warranted. Clusterin. APOJ, clusterin, is a 75 to 80 kDa dysfunctional linked heterodimetic protein associated with the clearance of cellular debris and apoptosis. In humans, cholesterol is encoded by the CLU gene on chromosome 8. Unlike most other chaperone proteins which aid intracellular proteins, CLU is a Golgi chaperone that facilitates the folding of secreted proteins in an ATP-independent way. The gene is highly conversed in species, and the protein is widely distributed in many tissues and organs, where it participates in a number of biological processes, including lipid transport, membrane recycling, cell adhesion, programming cell death, and complement-mediated cell lysis. APOJ peptide. Recently, there has been great interest in the use of apoprotein and APOA1 mimetic peptides as potential therapeutic agents for atherosclerosis. Low-density lipoprotein (LDL) and human APO1 are added together in a co-accumulation with human artery wall cells. The LDL is still able to stimulate the cells to produce MCA10. However, if APO1 is added to the artery wall cells removed before addition of LDL, or if APA1 is incubated with LDL and it can separate from the LDL before the addition of the LPO cells, APO gel also bound these inflammatory lipids so that they were inactive in a co-incubation, which means it could not incubate MCA. APOE deficient mouse. Apoprotein E is a class of apoprotein found in the cligomicron and intermediate density lipoproteins, IDLs, that is essential for the normal catalyzation of triglyceride-rich lipoprotein constituents. In peripheral tissues, APOE is primarily produced by the liver and macrophages and mediates cholesterol metabolism in an isoform-dependent manner. Knockout mice that lack the apoprotein E gene develop extreme hypocholesterolemia when fed a high-fat diet. Hypocholesterolemia, also called dyslipidemia, is the presence of high levels of cholesterol in the blood. It is a form of high blood lipids and hyperlipidemia and are associated with an increased risk of atherosclerosis and coronary heart disease. LDL receptor of a mouse. The low-density lipoprotein receptor is a mosaic protein of 839 amino acids that mediates the endocytosis of cholesterol-rich LDL. It is a cell surface receptor that recognizes the apoprotein B100, which is embedded in the outer phospholipid layer of LDL particles. The inhibition of macrophage ACYL coenzyme A, cholesterol activase ACAT, which catalyzes the formation of cholesterol esters, has been proposed as a strategy to reduce cell formation and treat atherosclerosis. We show here, however, that hypercholesterolemia LDL receptor deficient mice reconstituted with ACAT1 deficient macrophages, which unexpectedly develop larger atherosclerotic lesions than control mice. Lysophosphatic dolicholine (LPA). Lysophosphatic dolicholanes are also called lysolecithins, are a class of chemical compounds which are derived from phosphatidic dolicholanes. They result from partial hydrolysis of phosphatidylcholanes, which removes one of the fatty acid groups. The hydrolysis is generally the result of the enzymatic action of phospholipase A2. Among other properties, they activate endothelial cells during early atherosclerosis. Statins, also known as HMG-CoA reductive inhibitors, are a class of lipid-lowering medications that inhibit the enzyme HMG-CoA reductase, which plays a central role in the production of cholesterol. High cholesterol levels have been associated with cardiovascular disease. Statins have been found to reduce cardiovascular disease and mortality in those who are high risk. Statins inhibit action beyond lipid-lowering activity, the prevention of atherosclerosis. The ASTEROID trial showed different ultrasound evidence of arrhythmia regression during statin therapy. Researchers hypothesized that statins prevent cardiovascular disease. Lipitor. Atorvastatin, marketed under the name Lipitor among others, is a member of the drug class known as statins, which are also known primarily as a lipid-lowering agent and for prevention of events associated with cardiovascular disease. Like all statins, atorvastatin works by inhibiting HMG-CoA reductase as an enzyme found in liver tissue that plays a key role in production of cholesterol in the body. Zocor. Simvastatin, marketed under the trade name Zocor, among others, is a lipid-lowering medication. It is used along with exercise, diet, and weight loss to decrease elevated lipid levels. It is also used to decrease the risk of heart problems in those at high risk. It is taken by mouth. The primary uses of simvastatin are to treat dyslipidemia and to prevent atherosclerosis-related complications such as stroke and heart attacks in those who are at high risk. It is recommended to be used as an addition to a low cholesterol diet. Niacin. Niacin, also known as vitamin B and nicotinic acid, is an organic compound and, depending on the definition used, one of the 20 to 80 essential human nutrients. Pharmaceutical and supplemental niacin are primarily used to treat hypercholesterolemia, high cholesterol, and pellagra, niacin deficiency. Niacin has not been found to be useful in decreasing the risk of cardiovascular disease in those already on a statin, but appears to be effective in those not taking a statin. It reduces secondary outcomes associated with atherosclerosis, such as low-density lipoprotein cholesterol, LDL, very low-density lipoprotein cholesterol, VLDL-C, and triglycerides, TG, but also increases high-density lipoprotein cholesterol, HDL. Despite the importance of other cardiovascular risk factors, high HDL was associated with fewer cardiovascular events independent of LDL reduction. Pravastatin, marketed as Pravaco or Selcatine, is a member of the drug class of statins used in combination with diet, exercise, and weight loss for lowering cholesterol and preventing cardiovascular disease. Pravastatin is primarily used for the treatment of dyslipidemia and the prevention of cardiovascular disease. It is recommended to be used only after other measures such as diet, exercise, and weight reduction have not improved cholesterol levels. Aspirin, also known as acetylsalicylate acid, is a medication often used to treat pain, fever, and inflammation. Aspirin is also used long-term at low doses to help prevent heart attacks, stroke, and blood clot formation in people at high risk of developing blood clots. Low doses of aspirin are given immediately after a heart attack to prevent clotting and reduce the risk of another heart attack or the death of heart tissue. Aspirin is used as an anti-inflammatory agent for both acute and long-term inflammation, as well as for the treatment of inflammatory diseases such as rheumatoid arthritis. Zetia. Ezetimibe is a drug that lowers plasma cholesterol levels. It acts by decreasing cholesterol absorption in the small intestine. It may be used alone, or marketed as Zetia or Azotor, when other cholesterol-lowering medications are not tolerated, together with statins. Marked as Votorian and Energy when statins alone do not control cholesterol. While ezetimibe reduces LDL cholesterol, it has been shown to affect outcomes such as risk of death or major cardiovascular event like heart attack or stroke. Its effectiveness in reducing atherosclerosis in clinical trials has been inconsistent. Votorin. Ezetimibe and simvastatin is a drug combination used for the treatment of dyslipidemia. It is a combination of ezetimibe and the statin drug simvastatin. The combination preparation is marketed by Merck & Co. under the trade name Votorin in the U.S. and Igini in Europe. Ezetimibe reduces blood cholesterol by acting as the brush border of the small intestine and inhibiting the absorption of cholesterol, leading to a decrease in the delivery of intestinal cholesterol to the liver. Simvastatin is a HMG-CoA reductase inhibitor of statin. It works by blocking an enzyme that is necessary for the body to make cholesterol. Atherosclerosis. Atherosclerosis is a biopharmaceutical drug approved by the FDA on July 24, 2015. This is a second-line treatment for high cholesterol for adults whose cholesterol is not controlled by diet and statin treatment. Atherosclerosis is used as a second-line treatment to lower LDL cholesterol for adults who have severe form of hereditary high cholesterol and people with atherosclerosis who require additional lowering of LDL cholesterol when diet and statin treatment have not worked. Fish oil. Fish oil is oil derived from the tissues of oily fish. Fish oils contain the omega-3 fatty acids eicosapentaenoic acid and docosahexaenoic acid, precursors of current eicosanoids that are known to reduce inflammation in the body and have other health benefits. Nonetheless, fish oil supplement studies have failed to support claims of preventing heart attacks or strokes. New UCLA drug, APOA1 mimetic peptide. Apoprotein A1 is a naturally occurring mutated variant of the apoprotein A1 found in human HDL, the lipoprotein particle that carries cholesterol from tissues of the liver and associated with protection against cardiovascular disease. APOA1 has been shown to reduce atherosclerosis in animal models and in a small phase 2 human trial. Recombinant adeno-associated virus 8 mediated APOA1 gene therapy in combination with low cholesterol diet induces rapid and significant regression of atherosclerosis in mice.